Gene family is globin gene. A gene family is a set of several similar genes formed by duplication of a single original gene and generally with similar biochemical functions. It is a set of homologous genes within one organism. A family originates when a gene is duplicated. Initially, the two copies are identical, but then they diverge as mutations accumulate in them. Further duplications and divergence extend a family further. For example, globin genes with subfamily alpha globin and beta globin. When a gene is present in two or more copies per genome, this condition is known as redundancy. Members of a gene family may be either clustered together or dispersed on different chromosomes or present in a combination of both. A gene cluster is a group of two or more genes found within an organism's DNA that encode for similar polypeptides or proteins, which collectively share a generalized function and are located within a few thousand base pairs of each other. Globin genes encoding the various globin proteins, evolved from one common ancestral globin gene which duplicated and diverged about 450 to 500 million years ago. After the duplication events, differences between genes in globin gene arose from the accumulation of mutations. Ancestral globin genes, hemoglobin genes, myoglobin genes, and plant globin genes. And globin gene products hemoglobin gene, hemoglobin HB in our blood is produced by the hemoglobin beta gene HBB and the hemoglobin alpha gene HBA. These two genes are responsible for providing instructions for making the proteins beta globin and alpha globin. Beta globin and alpha globin are an important part of the larger protein called hemoglobin, which is found in the mammalian red blood cells. Adult human hemoglobin has 4 protein subunits, which consist of 2 subunits of beta globin and 2 subunits of alpha globin. The genes are coordinately turned on and turned off during the embryonic, fetal, and adult stages of development. The genes for alpha globin lie in the cluster on chromosome 16, while those of beta globin are located on chromosome 11. The alpha cluster is smaller, extends over 20 kb, and has 4 functional genes: alpha 1, alpha 2, zeta 1, and zeta 2, and 2 pseudogenes, psi alpha 1 and psi alpha 2. The alpha cluster is smaller, extends over about 20 kb, and has 4 functional areas: alpha 1, alpha 2, zeta 1, zeta 2, and 2 pseudogenes, psi alpha 1 and psi alpha 2. Zeta genes are expressed during the embryonic development. Alpha 1 and alpha 2 genes are expressed during the fetal and adult stages of development. And beta cluster consists over 50 kb and has 5 functional genes like epsilon, beta, delta, G gamma, and A gamma, and 1 pseudogene, psi beta. The two delta chains, the two gamma chains, G gamma and A gamma, differ from a single amino acid, glycine and alanine. Epsilon genes are expressed during the embryonic development. G gamma and A gamma genes are expressed during the fetal development. Delta and beta genes are expressed during the adult stages of development. An abnormal mutation in the globin gene can lead to a disruption of normal hemoglobin production. The two most common genetic blood disorders are sickle cell anemia and thalassemia. So, sickle cell anemia affects the shape of the red blood cells, leading to sickle cell-shaped cells that are sticky and clump together, causing painful blood clots and disrupting the flow of blood in the blood vessel. It also causes anemia due to low red blood cell count. Thalassemia also causes anemia as one of the globin genes, alpha or beta, is impaired in this disease. In alpha thalassemia, there are deficit synthesis of alpha chain. In beta thalassemia is caused by reduced adaptation synthesis of beta globin genes. Imbalances of globin genes cause hemolysis and impair erythropoiesis. Sickle cell anemia and thalassemia. Normal thalassemia, malformed red blood cell with a mutation on one of the two beta globin gene. A carrier is formed with lower protein production but enough hemoglobin. Without a mutation, enough hemoglobin, no thalassemia carrier. With one mutation, less hemoglobin, beta thalassemia carrier without illness but less hemoglobin, slight anemia. With two mutations, no beta globin, thalassemia, major patient with severe anemia. Thalassemia, alpha thalassemia, Beta thalassemia. Alpha thalassemia, 4 genes are involved in making the alpha hemoglobin gene. Alpha thalassemia major and alpha thalassemia minor. Alpha thalassemia major, mutated genes, moderate to severe manifestation. Then 4 mutated genes, rare, usually early death. But alpha thalassemia minor, 1 mutated gene, carrier of thalassemia, 2 mutated genes, alpha thalassemia trait. Then beta thalassemia, 2 genes are involved in making the beta hemoglobin gene. Beta thalassemia major and beta thalassemia minor. Beta thalassemia major in the sense 2 mutated genes, Cooley's anemia, mild to moderate manifestation. But beta thalassemia minor, 1 mutated gene, mild manifestation. Then myoglobin gene. Myoglobin is an iron and oxygen binding protein found in the muscle tissue of vertebrates in general and in almost all mammals. It is related to hemoglobin, which is the iron and oxygen binding protein in blood, especially in red blood cells. In humans, myoglobin is only found in bloodstream after muscle injury. Myoglobin is the primary oxygen carrying pigment of muscle tissue. High concentration of myoglobin in muscle cell allow organisms to hold their breath for a longer period of time. Diving mammals such as whales and seals have muscles with particularly high abundance of myoglobin. In humans, myoglobin is encoded by MB gene. Myoglobin is released from damaged muscle tissue which has very high concentrations of myoglobin. Myoglobin is a sensitive marker for muscle injury, making it a potential marker for heart attack in patients with chest pain. This shows the millions of years ago evolution. Ancestral globin, myoglobin, hemoglobin, alpha globin, beta globin, like that. Hemoglobin structure, myoglobin structure, and hemoglobin versus myoglobin structural differences. Myoglobin, 1 subunit, 1 heme group. But hemoglobin, 4 subunits, 4 heme groups. And cytoglobin gene. Cytoglobin is a protein product of CYGB, a human and mammalian gene. Cytoglobin is a globin molecule ubiquitously expressed in all tissues and mostly utilized in marine animals. It is thought to protect against hypoxia. The predicted function of cytoglobin is the transfer of oxygen from arterial blood to the brain. Like hemoglobin, it is the oxygen carrier and hemoprotein found in the nitrogen fixing root nodules of leguminous plants. It is produced by legumes in response to the roots being colonized by nitrogen fixing bacteria termed rhizobia as part of symbiotic interaction between plant and bacterium. Roots not colonized by rhizobium do not synthesize like hemoglobin. Like hemoglobin has close chemical and structural similarities to hemoglobin, and like hemoglobin it is red in color. Then organization of globin gene. Restriction enzyme mapping has been used to study the globin gene organization in normal and abnormal human DNA. Alpha and beta globin are most functionally significant globin. All globin genes have a common form of organization with 3 exons and 2 introns, suggesting that they are descended from the single ancestral gene. Both alpha and beta globin genes have 3 exons, and the 2 introns are located at constant positions relative to the coding sequence. The central exon represents the heme binding domain of the globin gene. Myoglobin is represented by a single gene in the human genome whose structure is essentially the same as that of the globin gene. Like hemoglobin gene contain 3 introns, the first and last which occur in points with coding sequence, and that homologous of these 2 locations is the 2 introns in the globin gene. The central intron of the hemoglobin separates 2 exons that together code for the sequence corresponding to the single central exon in globin. Evolution of globin genes. Examination of globin genes, whether from a mammal or a fish, reveals a characteristic organization. Each of the genes is constructed of 3 exons and 2 introns. Exons are parts of genes that code for amino acids and an encoded polypeptide, whereas introns do not. They are non-coding intervening sequences. Then examination of genes encoding certain globin-like polypeptides such as the plant protein like hemoglobin reveals the presence of 4 exons and 3 introns. This represents the ancestral form of globin gene. It is thought that the modern globin polypeptide arose from the ancestral form as the result of fusion of two of the globin genes some 800 million years ago. Some primitive fish have only a single type of globin gene, so they must have diverged from the line of evolution before the ancestral globin gene was duplicated to give rise to the alpha and beta variants. This appears to have occurred 500 million years ago during the evolution of bony fish. The next stage of evolution is represented by the state of the globin gene and the frog Xenopus levis, which has two globin clusters. The amphibian separated from the mammalian or avian line about 350 million years ago, so the separation of the alpha and beta genes must have resulted from a transposition in the mammalian or avian forerunner. After this time, there are separate clusters for alpha and beta globins in both birds and mammals. Thus, the alpha and beta genes must have been physically separated before the mammals and birds diverged from their common ancestor, an event that occurred probably 270 million years ago. Some other aspects of gene families. Protein coding genes may be solitary or belong to gene family. Solitary gene in multicellular organisms, roughly 20 to 25% of protein coding genes are represented only once in a haploid genome and are thus termed as solitary genes. Chicken lysozyme gene. It constitutes a single transcription unit and contains 4 exons and 3 introns. In gene families, definition and the concept. A gene family is a set of several similar genes formed by the duplication of a single original and generally with similar biological functions, or a set of duplicated genes that encode proteins with similar but non-identical amino acid sequences called a gene family. A set of duplicated genes that encode proteins with similar but non-identical amino acid sequences called a gene family. A gene family is a set of homologous genes within an organism. When a gene is represented in 2 or more copies per genome, the condition is known as redundancy. Members of a gene family may be either clustered together, dispersed at different chromosomes, or present as a combination of both. Genome analysis shows that many genes belong to families up to 25,000 genes identified in the human genome fall into 15,000 families. Genes are usually related to only by their exons, with introns having diverged. Molecular and nuclear organizers. Nucleus, the origin of the nucleus where the RNA synthesis occurs as a characteristic appearance with a core of fibrillar nature surrounded by a granular cortex. The fibrillar core is where the rRNA is transcribed from the DNA template, and a granular cortex is formed by the ribonucleoprotein particles into which the rRNA is assembled. The whole area is called the nucleolus. Here you could see structure of nucleolus. Nuclear organizers. The particular chromosomal regions associated with the nucleolus is called nuclear organized regions. Each nuclear organizer corresponds to a cluster of tandemly repeated rRNA genes on one chromosome. The concentration of tandem repeated rRNA genes and their very intensive transcription is responsible for creating characteristic morphology of the nuclei. Nuclear organized regions after silver nitrate staining using microwaves. Metaphase, the arrows indicate the NOR sites, interface and nuclei. Protein family. Encoded, closely related homologous proteins constitute a protein family. Its members may be clustered together or dispersed on different chromosomes. An extensive tandem repetition of gene may occur when the product is needed in usually large amounts. Genes for rRNA and histone proteins. Most protein families include from just a few to 30 or so members. Cytoskeleton proteins, 70 kDa, heat shock proteins, myosin heavy chain, chicken ovoalbumin, and alpha and beta globins in vertebrates. A few protein families include hundreds of members. Example, protein kinase, transcription factors, and vertebrate immunoglobulins. Then about gene cluster. A gene cluster is a part of a gene family. It is a group of 2 or more genes found within an organism's DNA that encode for similar polypeptides or proteins. They share generalized functions and are located within a few thousand base pairs of each other. The size of the gene cluster can vary significantly from a few new genes to several hundred genes. Gene cluster ranges from extremes in which duplication has generated two adjacent genes to case where hundreds of identical genes in a family. Gene found in a gene cluster may be observed near one another on the same chromosome or on different but homologous chromosomes. A multi-gene family is a member of a family of related proteins encoded by a set of similar genes. Multi-gene family is broadly used to include groups of genes from same organism that encode proteins from similar sequences either over their full lengths or limited to a specific domain. Multi-gene families are believed to have arisen by duplication and variation of a single ancestral gene, histidine, hemoglobin, immunoglobulins, interferons, tubulins, etc. DNA duplication can generate gene pairs. If both copies are maintained in subsequent generations, then a multi-gene family will exist. Multi-gene families are classified into two categories, like simple multi-gene family, organization of rRNA gene in Xenopus laevis, complex multi-gene family, organization histone gene in sea urchin, and organization of tRNA gene in Drosophila. The organization of rRNA gene in Xenopus laevis. Transcription units include the sequence of 18S, 5.8S, and 28S rRNA genes. The adjacent transcription units are separated by non-transcribed spacer regions called intergenic spacers, which separate adjacent rRNA genes in Xenopus laevis. Several sequence elements which include transcription terminators, region O repeats, region I repeats, duplicated spacer promoters, 42 base pair spacer elements. 42 base pair enhancer element which embedded within 60 or 81 base pair repeats. Thus, all have been reported to estimate RNA polymerase transcription from an adjacent gene promoter. The most portion of the intergenic spacer in Xenopus laevis consists of 31, 34, and 100 base pair repeats known as region O and region I respectively. Within each 60 to 81 base pair enhancer is a 42 base pair sequence. It is 80% identical to an upstream domain of the gene promoter. The enhancer evolved from duplicated promoter domains that bind essential transcription factors. Spacer promoters can program polymerase I transcription initiation, but their transcripts terminate upstream of the gene promoter at site T3. T3 is a fail-state termination site in that it prevents transcription from proceeding through the gene promoter. The function of spacer promoters in Xenopus laevis is not clear. In several studies, full enhancer function of 60 to 81% evidence. PP repeats are only realized in oocytes if at least one spacer is located upstream. This includes that the spacer promoter is needed for full enhancer function. Organism of histone genes in sea urchin. All histone genes are arranged in same order and orientation, although each is thought to be transcribed separately. The arrangement of genes indicate the possibility of common control of transcription. Although the overall size of clusters is different in each species, the relative separation of the gene is almost same. However, this appears to be no sequence relationship between the spacers of different species. Two different types of repeating units can be seen in sea urchins. Each has a single same length and arrangement of developmental variants of histone H1, H2B, H2A, and in sea urchin has been identified by direct analysis of proteins. Histone gene cluster of sea urchin. Organization of tRNA gene in Drosophila. Nucleotide sequencing of entire Drosophila tRNA coding region reveals 7 tRNA genes. 2 tRNA genes are nearly identical and containing intervening sequence of length of 38 and 45 bases respectively in the anticodon loop.